And now I would like to introduce, as I mentioned, Dr. Gary Pistano from Biodesics. He's going to speak to us on the Torrent Oncomine Dx Express test and the first experience in a CLIA CAP laboratory in the United States. So I'm sure we'll all be very, very interested in what he has to say. So I'll ask him to begin. Dr. Pistano.
Thank you, Bob. Thank you, Bob, for the kind introduction, and David for such a terrific lead-in to Oncomine DX Express test and the Genexus integrated sequencer. I'd also like to thank the Thermo team, of course, for the invitation to share our experiences in such an august forum. So a little bit about myself. First, disclosures. I'm also the New York State CLEP lab director for our laboratories, where at Biodesics, we really envision a world where all critical lung disease are diagnosed early and treated quickly. So many of the messages that David shared and that Thermo in general is focused on resonate very, very well with the Biodesics mission and vision. And actually, in point of fact, quite pleased with the first approval for EGFR exon 20 mutations in non-small cell lung cancer. So a little bit more about Biodesics and our clinical labs before we delve into data. We actually operate 2 certified high complexity laboratories. First, I'll start with introducing our laboratory here in Kansas, and many of those teammates have actually tuned in to this webinar today. This is also a CAP CLIA New York State CLEP certified laboratory that runs our nodule early cancer detection tests, and maybe can be a subject of a future webinar. But for today, I'm coming to you from our Colorado Louisville facility, where we recently moved in last year and a half. Bob, when you and I previously did some of these, we were in our Boulder laboratory, but we've established our CAP accredited CLIA CLEP certified laboratory here, which is also ISO 13485 certified. It's been our pleasure to work with the Thermo team. I think it's a 6-year or so experience to this point. And all of the studies I'll share today, and which are being prepared for publication, were conducted in the Colorado laboratory. The Oncomine DX Express test at the time, as a reminder, was investigational use only, so IUO. Same for the systems. And like I said, were conducted entirely here in our Colorado lab. So our objective, and I want to call out our team lead on this project, Amanda Weaver, who really worked with the lab resources on instrument installs and qualifications, et cetera, and to get with our external partners tissue specimens for which we had reference data that we could then do comparative assessment with ODX ET, Oncomine DX Express test. We obtained 48 individual FFPE specimens, 84 variants available for evaluation in the concordance study, 3 by 10 micron curls from, like I said, a commercial biobank. We used 1 curl in extraction with 2 as backup in case of repeats. I can share ahead, not many repeats were at all necessary. All specimens were extracted in the study on the Genexus system. As David outlined, all extracted nucleic acids were evaluated then with Oncomine DX Express Test IUO on the Genexus Dx integrated sequencers. We utilized 2 systems in this study, in part, of course, because we are preparing to make these tests and systems commercially available. That includes for clinical trial testing. So a part of our experience was also readiness and competencies within our laboratory to run the instruments as well as the assays. Now, the reference data, I don't know that today we have a lot of time, maybe in the Q&A, to go into the reference data, but this was independently generated by the partner biospecimen lab using a custom QIAGEN multimodal assay. This assay contains 64 DNA target genes, 6 RNA. Our receipt of information was limited to DNA. So for today, all of the concordance will be on DNA variants. but they were used as a reference for sample and variant level concordance. All of the reference results were blinded to our study operators at the beginning, during, and toward the end of the study. Confirmation of any first pass discrepant variants were with two alternative validated assays. And these are in-house validations. One is an amplicon-based NGS assay, and then the other comes from our droplet digital PCR validations, the GeneStrat test. So altogether, I would say not a small feat, tremendous undertaking by Amanda and the team. But in this concordance study now, you can, if you're keeping count, there are up to 4 different tests being used on multiple platforms to establish the user experience. But stepping back to laboratory experiences, just as a snapshot again, um, I'm happy to say that things worked out very well. But here are some of the user experiences, including mine. Very happy with the 2 touches. This was research associate who worked alongside Amanda to conduct the study. Um, Very convenient for our operators to have the same systems integrated in extraction and testing. Automation of the major steps of the NGS workflow, including sample purification, quantitation, library preparation, sequencing, bioinformatic analysis, and reporting are very welcome. And this is from our Director of Laboratory Operations. Remember, we today run what is a semi-automated NGS solution in our laboratory. So having the ability to really get feedback and compare the user experiences from the 2 separate systems, I think allows us to bring a unique perspective to this. Personally, I was very impressed with the fast turnaround time from sample loading to results. And David outlined how that workflow is implemented in a commercial laboratory. So our experience with sample processing, much like David shared, started with FFPE specimens onto Genexus Dx purification system, and then transferring of those plates onto the sequencer, really only 2 touches and generation of analysis and reporting at the end of the day 2. So again, really terrific user experiences. And we were able to process up to 12 samples, because again, we're doing a retrospective analysis at once, whether it's on the purification system or on the sequencer. So very high throughput at the same time we're getting rapid results. In terms of the samples, I know this is a little bit of an eye chart. Trust me, there was much more data on the demographics of the patients, but we decided to slim it down for this presentation. There are 13 cancers represented in this, so we really wanted to, not only what's the turnaround time, what's the ease of use, what's the concordance, but also really stress the system with many different cancers and different levels of variance, as you'll see, in terms of percent VAF representation. So 13 cancers, all selected deliberately by the availability of the reference data, as I said, from the QIAGEN multimodal custom assay to be available. So data in summary, and again, this is being prepared also for presentation, the CAP annual meeting, where we'll share more on the methods as well as what we did for discrepant reconciliation. But in summary, here we present the data at the variant level being very, very highly concordant. So 98% on the left, reference VAF versus ODX ET reported variant allele frequencies, and you can see a good representation of VAFs with really variance between the reference and the task over 84 variants, really at only 0.07%. Now, this is amazing. From my perspective, going into this, I would say hardly expected this level of concordance, not only at calling variants, but also the spread. Given the differences not only in extraction systems being used as a reference, um, purification systems, quantitation systems, sequencing, hybrid capture versus targeted amplicon, bioinformatics pipeline for calling, ultimately. On the right, post reconciliation, like I said, of any first pass discrepancies, we can see a very high degree of sensitivity and specificity, with 84 of the samples being called true positives. We had reconciliation of 5 specimens that were called with the QIAGEN multimodal, not with ODX-ET, but on reconciliation with orthogonal assays, we were able to show that ODX-ET indeed had the right call. So these were then not detected by ODX-ET on reconciliation. So in the end, currently we're at one purported false positive, one purported false negative, which we are in the process of still reconciling with these orthogonal technologies, NGS as well as droplet digital PCR. At the sample level as well, high degree of concordance. So we had 47 samples total eligible for reporting out of the 48. So one sample failed QC by pipette failure. And at the sample level, we achieved concordance then of 96%. At the variant level I just shared, 98% concordant with two variants still being reconciled. And they're in two different samples as well. So we look forward to updating, Bob, I would say maybe by the time of the CAP annual meeting on our final concordance. But right now, very good. So I would say bodes well, not only for clinical testing, but as well when we do our CAP proficiencies, I expect ODX-ET to perform exceedingly well. I'd like to thank our partners in the study, Thermo Fisher, as well as our team. This is a larger team representing here recently at Lab Week, and our mantra, which we really try to live here, of quality in your blood. So I'd like to thank everyone for the time and look forward to the Q&A. Over to you, Bob.
Thank you very much, Dr. Pistano. What a wonderful report from the first experience at a CLIA CAP laboratory in the United States. Again, very rich data and very clear data. I know we'll get into some of this questions about reference for variant levels, which seems to be a superbly elegant methodology that you deployed at Biodesix in this study. So my congratulations to you.